welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And as you can see on your screen, just for the chapter, Human Health and Diseases, and we are in part 1 of the series. All right, so let's begin. Okay, so question number 1. The infectious stage of Plasmodium that enters the human body is, and this appeared in NEET 2020, so it's pretty recent. A. Trophozoites, B. Sporozoites, C. Female gametocytes, and D. Male gametocytes. Okay, so to answer this question, you need to understand the life cycle of Plasmodium. And the answer is B. Sporozoites. Okay, so let's go to the discussion. Now, before I just, explain the answer directly, I thought that it would be important to explain the whole life cycle of Plasmodium and how it causes malaria. So I am now going to discuss the whole life cycle, okay, of Plasmodium. The life cycle of the malarial parasite. There are 4 species infecting humans. And those are Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale. Okay, so please remember these 4 names. Okay, so the genus is Plasmodium and the species are these 4. Okay, falciparum, vivax, malariae, and ovale. Okay, so in India, infections from Plasmodium vivax and Plasmodium falciparum are common. Okay. Now, the malarial parasite's life cycle includes 2 hosts. Okay, so there are 2 hosts that are involved. The first part is the asexual division, okay, the schizogony that occurs in humans, okay. So human is actually the intermediate host. Now, whenever you see the term intermediate host, it is the host where the asexual division takes place, okay. Sexual division does not take place in the intermediate host. It takes place in the definitive host, okay. And sexual development, that sporogony, occurs in female Anopheles mosquito, okay, which becomes the definitive host. So there are 2 hosts, okay, and through these 2 hosts, the malarial parasite completes its life cycle, okay. So there's an intermediate host where the asexual division takes place, and there is a definitive host where the sexual division takes place, okay. Please remember the genus name of the mosquito, that's Anopheles, and it is female, okay, not male. Please remember this, okay. It's the female that causes malaria. So let's move on. Now, what are the stages in human? Okay, now we know that there are 2 hosts, mosquito and the human host, right? So what happens in the human? Now, there are basically 4 stages that are happening. There's primary exoerythrocytic schizogony. Exoerythrocytic means it happens outside the RBC, the erythrocytes. There is erythrocytic schizogony. Then there's gametogony, where the male and the female gametes are formed. And there is a secondary exoerythrocytic schizogony, which is separate from the first 3. Okay, it happens at a later stage when there is relapse of malaria. Okay, so we'll come to every stage in detail. This I'm just explaining the names, what are the stages involved in humans. So the infective form of the parasite is sporozoites. We already discussed that. The spelling is wrong. It's sporozoites. Sorry about that. Okay, and are present in the salivary glands of the mosquito. Okay. Now, the sporozoites enter the bloodstream via the piercing of the skin by the proboscis. So if you look at the structure of the female mosquito, she has a proboscis, like a stick-like structure, like a nose, like a long nose, and she pierces into the skin. Now the sporozoites are present inside her salivary glands, and that goes into our bloodstream. Okay, that's when you have a mosquito bite. All right. Now, once the sporozoites enter the bloodstream, primary exoerythrocytic schizogony begins to happen. Okay, so sporozoites are actually elongated, spindle-shaped, and leave the bloodstream and enter the liver cells where they turn into round shaped. Okay, so they are in the bloodstream, they are long, elongated, spindle shaped, and they go into the liver cells, the hepatic cells, where they turn into round shaped. Now it undergoes multiple nuclear and cytoplasmic divisions and develop into primary exoerythrocytic schizont. Okay, so in the liver cells, it undergoes these multiple nuclear and cytoplasmic divisions, and they develop into schizont, primary exoerythrocytic schizont. And now remember, we are exoerythrocytic. Okay, so we are outside the red blood cells. Also, we'll come to the erythrocytic part, which is very important. It's the clinical, where we can see the clinical effect of malaria. Okay, but we'll come to that later. The schizont varies in size from 24 to 60 micrometers and contains 20,000 to 50,000 merozoites. Okay, so the schizont is this circular shaped and there are merozoites present inside. The liver cells rupture and release the merozoites into the bloodstream. Okay. Now, we finish the liver cell part. The merozoid, the schizont is formed, where the merozoites are formed inside, and that schizont ruptures and the merozoites entered into the bloodstream. Okay, so it took help of the host liver cells first. Okay, so now it has entered the bloodstream in the form of merozoites. The merozoites enter the bloodstream and they invade the erythrocytes. Okay, and multiply at the expense of the host cell. Okay, so this is where the dangerous part happens. Okay, it undergoes stages of trophozoites. So the merozoites convert into trophozoites, into schizons, and then again merozoites. Okay, so it undergoes a full life cycle inside the RBCs. Okay, so it is in the merozoite form, it invades the RBCs, again it converts into trophozoites, schizons, and then merozoites. Okay, depending on the species, 6 to 32 nuclei are formed which rupture the red cells, the RBCs, and release the merozoites and infect fresh erythrocytes. Okay, so now merozoites have entered the RBCs, they are undergoing the transformation into trophozoites, schizons, and then again merozoites. They rupture the RBCs and invade fresh RBCs, okay, that are not infected, uninfected ones. Okay, so when this rupture is happening and invasion is happening of fresh red blood cells, that's when the fever begins to occur. Okay, that's where you see the clinical part of malaria, which we will discuss this in detail later on. Okay, now gametogony. Now some merozoites develop into male. Now we saw that merozoites are infecting other RBCs, it's doing its job, but not all merozoites are focused on infecting other RBCs. Okay, so now what happens is some merozoites develop into male and female gametes. Okay, so sexual reproduction also has to take place for the Plasmodium to survive. Okay, so it needs gametes for that, correct? So it develops into male and female gametes. So the male are microgametocytes, I mean they're small in structure, and the female gametes are macrogametocytes. Okay, in the red cells of capillaries of internal organs like spleen and bone marrow. That means spleen and bone marrow also are being affected. The mature gametes are present in the peripheral blood. So if you want to look for mature gametes, you will find them in the peripheral blood. The immature ones that are growing up, you will find them in the red cells of capillaries of internal organs. Now the microgametes are small in size and macrogametes are large in size, as you can see from the name itself. Okay, so micro are the male, macro are the female gametes. Okay, now what is secondary exoerythrocytic schizogomy? Okay, so now what happens is, now in the case of Plasmodium vivax and Plasmodium ovale, sporozoites that enter the liver cells, not all undergo multiple nuclear division to form schizons. Remember when initially they form schizons, but not all of them form schizons. So what happens is they remain dormant in the liver cells for weeks, months, or even years. Okay, and they are known as hypnozoites. Okay, so basically they are dormant, they are asleep, they are not actively involved. Okay, not all sporozoites are going to form schizons. Some of them just remain in the liver cells and they remain inactive. Okay, so that is why it's called the secondary exoerythrocytic. Okay, it is secondary, it's not primary, and it remains in the dormant stage in the liver cells for even up to years, and they are known as hypnozoites. Okay, they are 4 to 6 micrometer in diameter and uninucleate. So the hypnozoites become reactivated and undergo division to form schizons and merozoites, which infect erythrocytes, causing relapse of malaria. Okay, so when hypnozoites become active, they undergo the same life cycle as what was happening initially in the first attack. So this is like a relapse of malaria. Okay, so this is like a reserve, like, so the Plasmodium or the protozoa is designed, the life cycle is designed in such a way that they have reserved some for later use. Okay, so whenever a relapse needs to happen, it will happen much later. Okay, it will not cause initial infection. Stages in mosquito. So now what we have seen are the 4 stages in humans, right? We saw that initially till the end we have seen that it has formed the male and the female gametes, right? Of course, it needs to continue its next generation, right? So the female and the male gametes will be formed, and they are formed in the erythrocytes. The sexual gametes are formed in the peripheral blood in humans and transferred to mosquitoes via blood meal. Okay, so whenever there's a blood meal where the mosquito pierces you, there'll be a blood meal where the mosquito will be able to suck in blood, right? And she sucks in blood, these gametes will also move into her system. So now when the gametes are sucked into her system, the gametes move into the stomach of the mosquito. So from one microgametocyte, 8 thread-like gametes form by the process of exflagellation. Okay, so the microgametocytes, they formed 8 thread-like gametes, but the macrogametes don't show any exflagellation. Okay, so what happens is, in the case of female gametes, the nucleus shifts to the surface where a projection is formed. Now when the projection is formed, when the projection is formed, fertilization occurs when the microgamete penetrates the projection, forming a zygote. Okay, so the fertilization is happening in the mosquito. If they ever ask you in the exam where the fertilization is happening, it's happening in the mosquito. Okay, it's not happening in the human. The gametes are formed in the human. You'll find the mature gametes in the peripheral blood. When the mosquito takes a blood meal from you, she will suck in the blood, right? So now the gametes have entered her system. Okay, where the microgametes form 8 thread-like structures, and the macrogametes don't perform any exflagellation. In fact, a projection-like structure appears, and where there'll be penetration of the microgamete with the female gamete, the macrogamete, okay, and resulting in fertilization. Now the zygote lengthens and matures to form ookinete. Okay, it forms ookinete, which is actually motile, vermiculate. Okay, it's a small motile-like structure. That means it moves, okay, and moves into the epithelial lining of the stomach, okay, and develops into an oocyst. Okay, develops into an oocyst. So first we saw how the female and the male gametes have matured. They are, being ready to perform the fertilization, and once the fertilization is performed, ookinete develops, okay, from the zygote, and it is actually a motile structure and moves into the epithelial lining of the stomach of the mosquito, which then develops into an oocyst, from ookinete into an oocyst. So the oocyst is rounded with a single nucleus and increases in size to a diameter of 40 to 50 micrometers, and inside this develops sporozoites. Okay, so inside the oocyst you will find sporozoites, which is the infective form. Now the number of sporozoites in each oocyst varies from few hundreds to few thousands. Now on the 10th day, oocyst fully matures and ruptures and releases sporozoites to various organs of the body, especially the salivary glands. So that's where the sporozoites then enter the salivary glands of the mosquito. So now the oocyst has a sporozoites, which is the infective form for us, okay, which causes malaria. Okay, so on the 10th day, the oocyst fully matures and ruptures. Okay, the sporozoites are released into the system. It goes to the other organs, but mostly settles there in the salivary glands of the mosquito. So hence at this stage is then capable of transmitting infection to man. So if the Plasmodium now has completed its full cycle, it has finished. We have seen that how basically our first stage was when it entered into the human system, right? And we have seen how it affects the human system, and then again goes back into the mosquito, and then again now it can infect the human again. The whole life cycle will repeat itself over and over again. Okay, so it's like a cycle which exists between man as in human and mosquitoes. Okay, the female mosquito, please remember that. So basically this is what the life cycle of Plasmodium is. I would suggest you can note these points down, so that's easy for you to study for the exam, and if they can ask, if they ever ask any question, you can answer the question very easily. Okay, it's not very difficult. Okay, if you keep reading it over and over again, you'll remember it. Okay, so this is the different species of Plasmodium and the incubation period. Okay, this can also be a potential NEET question if they ever ask you what is the incubation period of falciparum or vivax or malaria or whatever. All right, so learn this table for your sake, for the exam's sake. All right, now the stages of fever. Now, remember when I spoke about the erythrocytic stage? So there are the merozoites inside that, and they rupture the red cells and they infect other fresh red cells, right? So red blood cells. So that keeps happening, in circles. It keeps happening over and over again. Okay, so the stages of fever, this fever system is also like a cycle. Okay, so it begins in the early afternoon and is divided into 3 stages: cold stage, hot stage, and sweating stage. Okay, so if you notice that a fever in a patient is happening during this time, there's a possibility that this person has malaria. Okay, it's divided into 3 stages: cold stage, hot stage, and sweating stage. Now, the cold stage lasts for 15 to 60 minutes. Hot stage lasts for 2 to 6 hours, and the fever ends with profuse sweating. Okay, so this is the cycle that it adopts. Okay, the fever cycle that it adopts. So every time there's breakage or rupture of red cells and infecting new cells, there'll be this the stage of fever. Okay, where there is cold stage, there's hot stage, and then there's sweating. Then again there's cold stage, hot stage, and sweating, and again, and it specifically begins in the early afternoon. Also, depending on the species, the fever also varies. Okay, so if it is Plasmodium vivax, it demonstrates benign tertian fever. So remember this for NEET. Okay, it's a possibility that a question like that would also appear. Okay, Plasmodium vivax demonstrates benign tertian fever. Okay, and fever occurs once in 48 hours. Okay. P. ovale demonstrates ovale tertian fever. Again, the fever occurs in 48 hours. So where you see the word tertian, it means the fever occurs in 48 hours. All right. P. malariae demonstrates quartant fever. Okay, the fever occurs once in 72 hours. Okay, so where you see the word quartant, and it you have to remember this. Okay, which species and what kind of fever happens during with which species. You'll have to remember the names. There's no other way out. Okay, but for once I can give you a hint that whenever you see the word quartant, it's once in 72 hours. When you see tertian, it's once in 48 hours. Now, falciparum is different from the other 3. Okay, falciparum demonstrates malignant tertian fever. Okay, when you see malignant tertian fever, the fever is prolonged and intense. Okay, it's not like once in 48 hours or 72 hours. It's prolonged and intense. Now, anemia develops due to destruction of infected erythrocytes, obviously, okay, because you see that it attacks the red blood cells and ruptures. Again, it attacks new red blood cells, rupture. So the red blood cells are being destroyed, and obviously that will result in anemia. Okay, and there's reduced erythropoiesis in bone marrow and lysis and phagocytosis of uninfected erythrocytes. The splenomegaly is due to proliferation of macrophages that phagocytized parasitic and non-parasitic erythrocytes. So splenomegaly is spleen enlarges in size due to this. So that's another effect of malaria. The treatment of malaria is chloroquine was the standard treatment till pathogens became more resistant to it. Right, so quinine is the most reliable alternative. There's tetracycline and clindamycin exhibit anti-malarial treatment. Okay, so this is what's used currently for treatment of malaria. Prevention: the use of larvivorous fish Gambusia affinans, and there's a bacterium called Bacillus thuringiensis, which is a spore forming toxic protein. Okay, so which when ingested by mosquito larvae, the mouth parts and guts are paralyzed and the gut epithelium is destroyed, okay, leading to death. Okay, so there is a fish which feeds on larva. Okay, so that becomes easier for you to prevent the proliferation of larvae in stagnant water. Especially in stagnant water, malaria is an issue. Okay, and also there's a bacterium called Bacillus thuringiensis, which is a spore forming bacteria, and that is when it is ingested by the mosquito larvae, it can kill the larvae. Okay, so here is a diagram of the life cycle of Plasmodium. So go through this, okay, and I've taken this from a book called Medical Parasitology. Don't bother looking into it, I'm just telling you for your reference. Just go through the diagram here. Okay, and I think there is a diagram of the life cycle in NCERT too. Please go through that NCERT. You have to read without any doubt. All right, this is just for your reference. All right, so basically we've answered the question. It was Protozoa is the infective form of Plasmodium, which eventually causes malaria. And now, since I've explained the whole life cycle of malaria, you can note it down in your notebooks and read it over and over again, because the more you read, the more you will be able to remember the information. Okay, so if they ever ask any question related to life cycle of malaria, you can answer it very easily. Okay, so clear, let's move on. Okay, so question number 2. Match the following diseases with the causative organism and select the correct option. This appeared in NEET 2020. You have column 1 and column 2. Let's match the following. Typhoid is Salmonella. Pneumonia is Haemophilus. Filariasis is Wuchereria. And malaria is Plasmodium, which we just discussed. So it's A3. Okay, A3 here. You have a B4. You have a C1. You have D2. So answer is B. Yeah, it's B. Okay, let's go to the discussion. So here is the table for the answer that the question that we just saw. Note this down. Okay, one more thing I just wanted to clarify here. It's Wuchereria bancrofti, yes, and there's another species called Malai also, which causes a filariasis, but in certain like medical parasitology textbooks they have written as Brugia. Okay, so, and in NCERT it says W. Malai. Learn it as it is in NCERT, so it doesn't cause any confusion, but it also in medical textbooks they use the genus Brugia Malai. All right, okay. So let's talk about typhoid in a little detail, because these diseases have been briefly discussed in NCERT. That is why I'm discussing this here. Because there are endless lists of diseases. Okay, there are parasites and bacteria and viral diseases, and so on and on and on. A tapeworm and roundworm, and it will go on, and it's like a never-ending saga, right? So I can't like discuss everything. But the ones that have been discussed in NCERT, I'm discussing it in a little more detail. Obviously, please read NCERT, the information there, and you can also come back to the discussion after that. So it's caused by Salmonella typhi. So it can spread by foodborne or waterborne transmission from fecal contamination by ill or asymptomatic carrier. Symptoms include fever, which occurs in more than 75% of cases. This abdominal pain, which is 30 to 40%. The incubation period is on an average of 10 to 14 days. Okay. The prominent symptom is prolonged fever, which can continue up to 4 weeks. The early physical findings include rash. You'll find rash. You'll find hepatosplenomegaly, epistaxis, which is nosebleeds, bradycardia at the peak of high fever. So these are some of the physical findings that you'll see in the case of typhoid. Severe cases include gastrointestinal bleeding and intestinal perforation in the 3rd and the 4th week of illness. All right, so go through this, go through the notes in NCERT, and you can combine all the information and piece it all out together. Okay, what is typhoid and what are the symptoms and what are the physical findings that you find in typhoid. Okay, so if they ever ask you a question related to this, you can answer it easily. Treatment include antibiotic therapy. Right. So we've just gone through typhoid. Let's go through pneumonia. Now, most commonly caused by Haemophilus influenzae and Streptococcus pneumoniae. Now, please remember, if you see the word influenzae, it has nothing to do with the influenza virus. Okay, that's a viral disease. Okay, and this is different. So please don't confuse the two. Okay, if you see Haemophilus influenzae, it is associated with pneumonia. Okay, and Streptococcus pneumoniae, yes, from the word itself, pneumonia, you can understand that it causes pneumonia. Okay, so please don't confuse the two. So the pneumococci colonizes the nasopharynx and the pathogens are then carried to adjacent areas. Okay, that's the eustachian tube and the nasal sinus. So it infects all these areas. This prevents clearance of bacteria. Okay, so now once the pneumococci infects or colonizes all these areas, you can't, you feel your area, your nasal areas or sinuses all feel blocked. Okay, so because of that, and so you can't clear out all the bacteria that's there. So pneumococci are resistant to phagocytosis. Okay, that's an interesting point. And pneumonia ensues when the pathogen is inhaled or aspirated into the bronchioles and alveoli and are not cleared. Okay, so they are resistant to phagocytosis, and the pneumonia then moves into the lungs. Okay, the bronchioles and the alveoli, and they're not cleared from there. Now, this could occur due to increased mucus production or damage to ciliary action by viral infection. The symptoms include inflammatory response such as pain by increasing pressure, may prevent oxygenation of blood or inhibit blood flow. Now, pneumonia becomes a very dangerous situation because of these reasons. Okay, there is inflammatory response, there's pressure, increased pressure. It prevents oxygenation of blood and inhibits the blood flow. Also, this cough and sputum production, this fever. Then you can even see through the X-ray the radiographic detection of infection. All right. Now, pneumonia is treated via beta-lactam antibiotics. Okay. So, Streptococcus pneumonia is the reason where all this occurs. We'll also discuss Haemophilus influenzae and what happens. That is mostly similar to Streptococcus. We'll see that. Now, when we talk about influenza, Haemophilus influenzae, there are 6 types, okay, that have been identified, and they're exclusively human pathogen. That's an interesting point to remember. H. influenzae type B causes disease in infants and children less than 6 years of age. Okay, it causes pneumonia, which infects the pleura, epiglottitis, and cellulitis in children. It is spread by airborne droplets or by direct contact with secretions and fomites. Fomites is when there are material structures which contain these pathogens, and you touch them and you touch your face. So that's how spread happens. Okay, so it can be spread through that also. Now they invade the respiratory system and spread to distant sites such as meninges, joints, and bones. Okay, so yes, Haemophilus influenza has the ability to do that. Okay, it not only affects the respiratory system, but it also damages the meninges, joints, and bones. The type B polysaccharide capsule prevents opsonization, allowing for it to cause systemic disease. Okay, so you see the type B Okay, so type B is a type of polysaccharide capsule that covers it. Okay, and it prevents the opsonization. Opsonization is the method by which opsonin attacks the pathogens, which we will study later on when we study antibodies. Okay, and the immune system. So don't worry too much on that now, for now. So it prevents that. Okay, so it's like a protective layer. The symptoms include a fever, cough, and purulent sputum. Vaccines are available for Haemophilus, and cephalosporin drugs are used to treat meningitis. All right, so these are the two pathogens by which pneumonia occurs. Okay, so and I also told you influenza does not mean it causes influenza. That's a viral disease, right? Okay, so we've gone through typhoid, we've gone through pneumonia. Let's look at filariasis. There are 4 species that infect humans: is Bancrofti, Malay, L. loa, and Onchocerca volvulus. Okay, they're very long names. And this species belong to the phylum Nematodes. So it occurs in Asia, Africa, Australia, Pacific, and South America. Now, the adult male and female worms reside in the lymph nodes and the lymphatic vessels of humans, and the microfilariae, that's the embryos, reside in the peripheral blood. So filariasis basically just attacks the lymphatic system. Okay, remember it that way. There is no doubt about that. It attacks the lymph nodes, it resides there, and the lymphatic vessels of the humans. Let's look at the life cycle of Bancrofti. Now, there are 2 hosts in its life cycle. Man is the definitive host, and female mosquito is the intermediate host. Okay, so a Culex mosquito is the most important vector. All right. Now, there are other Aedes and Anopheles also genus, but Culex is the most common vector that is found, which carries this pathogen. The adult worms reside in lymph nodes and lymphatics of the inguinal, scrotal, and abdominal. That's the lower limb area. All right. Male fertilizes the female, hence giving birth to microfilariae. They travel to the peripheral circulation. Now, the microfilariae appear for 2 hours at night and then remain in the pulmonary circulation. Okay, that's how the embryo functions. Okay, it appears for 2 hours at night and then it remains in the pulmonary circulation. So if you look at the peripheral blood, you will not find it there, but you'll find it only for those 2 hours at night. Okay, they are then ingested by the mosquito during a blood meal and reach the stomach. Their hyaline sheath are casted off in 2 to 6 hours and they penetrate the stomach wall. Now, in 4 to 7 hours, they reach the thoracic muscles, and in 2 days, metamorphose into short sausage-shaped organism with short spiky tail. So, this is the first stage larva. All right. And 3 to 7 days, they mold to become second stage larva. Right. So, all this is happening inside the mosquito. On 10th and 11th day, the larva metamorphosis is completed and the tail atrophies and the digestive system, body cavity, and genital organs are fully developed. This is the 3rd stage larva, most infective stage. This is what I wanted to come to. If they ever ask in NEET, if they ever wanted to mess around with you and they ask the most obscure question, ask which is the most infective stage in which area of Bancrofti, it's the 3rd stage larva. Okay, so that is why it is important to understand the life cycle of these pathogens, right? To understand which is the infective form of that particular pathogen. So this is the 3rd stage larva. It's the most infective stage. The larva migrates from the thoracic muscles to the proboscis of the mosquito and enter the human through the punctured skin. Now this is more or less similar to malaria. Not similar in the sense there are 2 hosts. There's a mosquito and then there's a human host, right? Obviously this is different from malaria. It functions differently, and it's also the pathogen is different. It's a nematode. So yeah, please go through this, write it down, read it over and over again to remember it, see what is happening in the human, what is happening in the mosquito. But like I said, go through NCRT, that is priority. Use this as a support. Okay, use these notes as a support to understand how filariasis takes place. So if they ever ask you any question related to filariasis, you can answer it easily, right? Like they say, which is the infective, which larva stage is the most infective form, right? So if you knew the life cycle, you would have been able to answer it very easily, right? Which is the most common, the vector that carries this pathogen. It's Culex mosquito. These questions are something that would they can ask easily in NEET if they want to up the game and make it a little difficult. So yeah, so once they enter the human through the punctured skin, they settle in the lymphatic system where they mature into adult worms and fertilize to produce microfilariae, thus repeating the cycle. So the lymph nodes become enlarged, firm and fibrotic. Because of that, the permeability of lymph walls cause leakage of fluid with high concentration of protein into the surrounding tissue. The leakage of lymph into the tissue results in lymphoedema. Okay, so that's where the swelling happens. The elephantiasis of one or more limbs, breast, penis, scrotum, or vulva happens. The treatment include administration of diethyl carbamazine orally for 12 days. Okay, so that's the treatment used. Sorry, I don't know if I'm allowed to show you photos of elephantiasis, but I think if you just do a quick Google search, you'll see how the lower limbs, the feet, the legs and all are enlarged when there's an infection of filariasis. All right. So this is basically filariasis for you. This I've just explained briefly the life cycle for you to understand how this actually works. Okay. So yeah, this is the life cycle of Wuchereria bancrofti. Oops, sorry. Okay, so go through this, compare it to the notes for you to just understand how the life cycle works. All right, so I think we've discussed, we've answered all the questions there, right? The question number 2, that was. So I have put the particular pathogen there and the disease alongside that. Please remember it, all right? And so that you know. Okay, so question number 3. Identify the correct pair representing the causative agent of typhoid fever and the confirmatory test for typhoid. Okay, this appeared in NEET 2019. You have a Salmonella typhi, that's Vidal test, B Plasmodium vivax UTI test, C Streptococcus pneumoniae Vidal test, and D Salmonella typhi anthron test. So typhoid fever is caused by Salmonella typhi, which is obvious, we already know that. The confirmatory test for typhoid is Vidal test. Okay, so the answer is A. The answer over here is A. The causative agent for typhoid fever is Salmonella typhi, and the confirmatory test is Vidal test. It is an agglutination test, okay, where the number of antibodies that are attached to the antigens present in the serum of the patient is measured. Okay, so it's an agglutination test. Okay, it's like an ELISA. All right, so please remember this, again factual based. It's there in NCERT also, so very important with respect to your exam. All right, so question number 4. In which disease does mosquito transmitted pathogen cause chronic inflammation of lymphatic vessels? We just discussed that. This appeared in NEET 2018. So it causes chronic inflammation of lymphatic vessels. Okay, so we have A elephantiasis, B Ascariasis, C ringworm disease, and D Amibiasis. Okay, so it's clearly elephantiasis. Answer is A. Yeah, so chronic inflammation of lymphatic vessels is caused by elephantiasis. Now, yeah, that is obviously the direct answer, but I'm also going to discuss the other diseases that are there mentioned in the answer. So let's go through Ascariasis. It is caused by Ascaris lumbricoides. Okay, the adult worms reside in the small intestine of humans. That's the jejunum. In the life cycle of Ascaris, there's only one host, which is humans. Okay, the fertilized eggs are passed in the feces. They reside in the soil and at the optimum temperature and humidity, a rhabdity-form larva develops from the unsegmented ovum. Okay, so the fertilized eggs are passed on. So the man is infected, the human is infected, and it passes the eggs through the feces. Okay, and that's where the larva is formed in these unsegmented ovum. The ovum is called unsegmented ovum. That undergoes first molting within the eggshell in 10 to 40 days. The eggs containing the larva are pathogenic. That's where the problem is. Okay, the eggs containing the larva are pathogenic. Now, when humans ingest food, water, or raw vegetables contaminated with larval eggs, they enter the small intestine and the ingested eggs hatch to liberate the larvae. Okay, so it is very important that you clean the vegetables that you eat properly. The food is not contaminated, the water is not contaminated, because they can contain that larva. Okay, so now when these larval eggs are consumed, they enter the intestine, the small intestine, and the ingested eggs hatch to liberate the larvae. Now, they enter the mucous membrane of the small intestine and enter the liver via portal circulation, where they reside for 3 to 4 days. They travel through the hepatic vein, inferior vena cava, right heart, and pulmonary artery and reach the lungs. Okay, so it goes through the whole procedure and enters the lungs. Okay, so they travel through the hepatic vein, the inferior vena cava, the right heart, the pulmonary circulation, the pulmonary artery, and then reach the lungs. Now in the lungs, they increase in size and molt twice. That's 5th and the 10th day. Okay, so the first molting has happened there. Now this second molting has happened the 5th and the 10th day. Twice it happens in the lungs. Okay, the larvae break through the capillary wall and reach the lung alveoli. From there, they migrate up the bronchi, trachea, and larynx over the epiglottis, pharynx, and swallowed and enter the upper part of the small intestine. So it entered the small intestine, it has again gone through the liver, up through the heart and into the lungs, and then up into the pharynx, and then again got through the epiglottis into the esophagus, back into the small intestine. Okay, where it has molted through the period. Okay, it has grown in size, it has molted there, in the lungs, and again it has gone up. So it has gone through this whole cycle inside your body. Now, on the 25th to 29th day, the larva undergoes another molting and transforms into adult worms. So it has taken all this time to grow in size, and again it has gone back into the small intestine, where by the 25th to 29th day, the larva undergoes another molting and they transform into adult worms. Okay, so now they have to become sexually mature. The whole purpose of pathogens is to form the next generation, right? So they have to sexually reproduce. So in 6 to 10 weeks, they become sexually mature, and by 12 weeks, the female begins to discharge eggs in the stools, thus repeating the cycle. So again, the female lays the eggs, and again they are passed through the stool as by the feces into the environment, and again the whole cycle repeats itself. Okay, so it is undergoing a lot of molting, and through the whole body it goes back into the intestine, where again it matures, and then again the whole cycle repeats. The nutritional status is affected, leading to malnutrition, that's malnourishment, and night blindness also. Night blindness due to deficiency of vitamin A. Okay, so there's deficiency of vitamin A in this, leading to night blindness. Okay, the presence of worms leads to cramps and loss of appetite. Now, the adult worm release a toxin called ascaris, which causes fever, wheezing, and conjunctivitis. So what you see here is the life cycle of Ascaris. Okay, so you can go through this. The treatment includes administration of pyrantel, pamoate, mebendazole, and piperazine. Okay, so these are the medicines that are used for treatment of Ascaris. Okay, so Ascaris is dangerous. So that is why it is important to clean, to wash, to make sure you're drinking not contaminated water, you're drinking eating non-contaminated food, so it is important to be careful. All right, so we discussed Ascaris. Let's go to Amibiasis. It is commonly found in tropical and subtropical countries. And Entamoeba histolytica exists in 3 morphological states. Okay, remember there are 3 different states in which it exists. There's trophozoite, there's pre-cyst, and there's a cyst, which is uninucleate, binucleate, and quadrinucleate. The trophozoites reside in the mucosa and submucosa of large intestine in man. What is the life cycle of that? The life cycle is in one host, which is human. It is ingested through contaminated food and water. Again, this is the same thing. You avoid contaminated food and water, you're not going to get infected. And then if you have or eat contaminated food and water, there is a chance that you could get an infection. Okay, so that's why it's very important. So it is ingested through contaminated food and water with mature quadrinucleate cysts. Okay, so mature quadrinucleate cysts is what enters into the humans. Then the intestine, the cyst wall is digested by trypsin, and a single tetranucleate amoeba metacyst is liberated. Now each nuclei divide by binary fission, forming 8 nuclei, and this gives rise to 8 mature cysts. That's amibulae. Okay, now this process is known as excistation. So initially excistation is the rupture of the cyst. Okay, so we saw that the cyst entered our system, the human system, and with the help of trypsin, it was liberated. The cyst wall broke and it was liberated. Okay, and these nuclei were divided by binary fission. Okay, forming 8 nuclei. And then this gave rise to 8 mature cyst ambulae. Oh, they're actually known as metacystic trophozoites. So this is known as excistation. Now they are carried into the cecum and invade the mucosa and live in the submucosa of the large intestine. Okay, so they follow the path of the stool formation, the feces formation. Okay, so they go into the large intestine, they go into the cecum, and they live in the mucosa and the submucosa of the large intestine. Now they grow and multiply by binary fission. Now Entamoeba histolytica secretes a proteolytic enzyme called histolysin. Okay, which destroys and causes necrosis of tissue and produces flask-shaped ulcers. Okay, so when it resides in the intestine, large intestine, it releases this histolysin, or this enzyme, okay, which destroys the tissues that are there in the intestine. Now, at this stage, a large number of trophozoites are excreted along with blood and mucus, leading to amoebic dysentery. So dysentery is not just diarrhea, it's when the stool is removed out, and there's blood found in it. So these trophozoites is called dysentery, basically amoebic dysentery. There's also bacillic dysentery, we'll come to that later. This what we are seeing is amoebic dysentery. Okay, so at this stage a large number of trophozoites is another form in which Entamoeba histolytica exists. They are formed and they are excreted along with blood and the mucus, leading to amoebic dysentery. Now, after the effect of the parasite is reduced, the patient passes normal stool and the trophozoite discharge undigested food particles and transform into pre-cysts and then into mature quadrinucleate cysts. Okay, so it undergoes this whole cycle. Trophozoite again gets transformed into cysts and then into, pre-cysts and then into cysts. Now, quadrinucleate cysts are the infective form of parasites. Again, this is a potential NCERT question. Okay, if they ever ask you which is the infective form of Entamoeba, so it's quadrinucleate cysts. Okay, they are the infective form of parasites, and this process is known as incystation. We saw excistation where the cyst wall ruptured, right? Incystation, the cyst is formed and it remains there. Okay, it remains there till there is another source by which the rupture of the cyst wall will happen. Okay, till then it preserves itself and it remains there in that cyst form. Okay, that's incystation. So quadrinucleate cysts are the infective form of the parasites. The treatment include use of amebicides and replacement of fluid, electrolytes and blood. Okay. So yeah, this is it. So yeah, this is the life cycle of Entamoeba histolytica. So you see the excistation part here, okay, where the cyst wall has ruptured, and here is the formation of the cyst, the quadrinucleate cyst. Okay, so the cycle keeps repeating itself over and over again. Okay, go through it. Like I said, read NCERT first and then come back to these notes, and you can note them down if you like. Okay, for your information, I would suggest specifically remember the infective forms of every pathogen, what type of pathogen it is, whether it's a nematode, whether it's an amoeba, whatever the case is. Okay, and it's a protozoa, so on, and that is important to remember. All right. Ringworm disease, and I spoke about Ascaris and we spoke about Amibiasis. It's a ringworm disease. Ringworm is caused by fungal genus Taenia. So it's a fungal disease. Okay, it's a dermatophytic fungi that releases proteolytic enzymes and keratinases into the skin. Okay, so it affects the skin. They release nutrients and disseminates through the stratum corneum. Okay, so stratum corneum is the upper structure of the skin, and they release the nutrients and this spreads on the stratum corneum. Now there's appearance of lesions, scaly patches with blood filled with vesicular borders and clear in the middle. So you have a reddish patch around and the middle would be clear. Okay, so you have blood filled vesicular borders and it's clear in the middle. Okay, so this is what a ringworm disease looks like. Now, just get rid of this. Taenia corporis causes ringworm disease of the skin. So they ever ask you in the exam which pathogen, which fungus causes ringworm disease of the skin. It's Taenia corporis, and Taenia pedis causes infection of the feet. If they ever ask you the fungal infection of feet, it's Taenia pedis. And treatment include topical agents. Okay, so there are creams that you can apply on the surface of the skin to heal the ringworm disease. Okay, so now we have seen a fungal infection. Okay, so that is why I said that please remember which pathogen causes which disease, and whether it's a viral disease, it's a bacterial disease, it's a protozoal disease, whether it's an amoebic disease, whether it's a fungal disease, whatever the case is. Okay, so you need to remember which is what. And of course, in the case of NCERT, they have discussed some of these diseases, not so much in detail except for malaria. Yes, malaria is important. It's a very common problem in India also. So understanding the life cycle of Plasmodium is very important. That is very, very, very important. That's like 3 stars, to that. Apart from that, I have discussed the other life cycles also of Ascaris, Amibiasis, Filariasis, and so on. So you can go through that. Remember what are the infective stages. So they ever ask you the infective stages, you just know on the top of your head, yes, this is it. Okay, and yeah, let's move on. All right, so question number 5: which of the following sets of disease is caused by bacteria? Okay, so yeah, so you need to know which of them is a bacterial disease, and this appeared in NEET 2016. So you have A, cholera and tetanus, B, typhoid and smallpox, C, tetanus and mumps, D, herpes and influenza. So clearly it is A, cholera and tetanus are both bacterial diseases. So your answer is A. Okay, so here you have table. Please note this down for your information, which is the causative agent for every disease and whether it's bacterial or virus. I have already mentioned that in the brackets over here, right? So pretty direct answer, a direct question, direct answer. You just need to remember this, and the more and more you read, the more you'll remember it. All right, let's move on then. Okay, question number 6. Which of the following disease is caused by protozoan? So, and this appeared in 2015. You have A, babesiosis, you have B, blastomycosis, C, syphilis, and D, influenza. Okay, so it has to be a protozoan disease. So it can't be B, it can't be syphilis, no, and influenza, no. So it's babesiosis. So the answer is A. Let's go to the answer discussion. So yes, babesiosis. These are the three parasitic protozoans that cause babesiosis. Okay, so please note them down. You have blastomycosis. That's a fungal disease. When you look at the word mycosis, you know it's a fungal disease, right? So you can differentiate that from babesiosis. Syphilis, we have already discussed. Treponema pallidum. Please check my videos on reproductive health. I've discussed syphilis in very much detail. Influenza virus. It's not Haemophilus influenzae. I have already discussed that also. So please don't get confused. All right. Okay. Let's move on then. Okay. So question number 7. Match each disease with its correct type of vaccine. And this appeared in 2015. It's a cancelled exam. All right. So tuberculosis, whooping cough, diphtheria, and polio. So diphtheria is actually inactivated toxin. It's a type of vaccine which contains an inactivated toxin. Polio is harmless virus. Tuberculosis is so I think killed bacteria. A3. No, it has to be A4. I'm sorry. A4 and has to be B3. Whooping cough. That's pertussis. All right. So A4, B3, C2, and D1. So the answer should be A. Okay. For the discussion. Okay, so I have made a table here. So please learn this table. It'll be easier for you to answer the question. So tuberculosis contains harmless bacteria. Now there's a difference between harmless and killed bacteria. So when you look at the word harmless, it means that the bacteria is attenuated. That means it's weakened, okay, in the vaccine. Now vaccines are designed in such a way that they instigate the immune system, to become active against that particular pathogen. Okay, so the vaccine is designed, in such a way that does not cause you infection. It should not cause you infection because of the vaccine, right? But only made in such a way that it is stimulating your immune system to create antibodies against that particular pathogen, right? So when now in the case of tuberculosis, they have made a harmless bacteria which is attenuated, which is weakened. Okay, now when they say killed bacteria, the bacteria is killed, it's inactivated. What they call is inactivated. Okay, that's a killed bacteria. Now in the case of whooping cough, which is called pertussis, I've also mentioned the causative agents, so what is causing every disease, each disease that's mentioned here. So that's Bordetella pertussis, tuberculosis, Mycobacterium tuberculosis. That's a harmless bacteria. This is a killed bacteria which is inactivated. Now diphtheria is Corynebacterium diphtheria. It's an inactivated toxin. Okay, so diphtheria actually produces a toxin. So now to counteract that, we don't want to vaccinate somebody with the active toxin. So it has to be inactivated, right? But that only is used to stimulate the immune system to attack the pathogen when it is there in the body. So it's an inactivated toxin. And polio, there are two types of polio vaccines. There's a harmless virus called Sabin polio vaccine. That's harmless is attenuated, of course, and inactivated polio vaccine, Salk polio vaccine. So please learn these names, Sabin polio vaccine and Salk polio vaccine, so you know which is what. Okay, so Sabin polio vaccine is a harmless virus, Salk is an inactivated polio vaccine. Okay, so these are the two types of polio vaccines that are again present there against the virus. All right, so learn this table for the exam, will be helpful. Okay, let's move on then. Question number 8. The active form of Entamoeba histolytica feeds upon, we already discussed Entamoeba histolytica, right? 2015 cancelled. Okay, so A, food in intestine, B, blood only, C, erythrocytes, mucosa and submucosa of colon, D, mucosa and submucosa of colon only. So we know that Entamoeba histolytica resides in the large intestine. Okay, it enters through the cecum and resides there. So we know mucosa and submucosa of colon is there, but which is the active form? The active form is the trophozoites, okay, which are active, which move around. Okay, and they also reside in the erythrocytes. So there's blood also coming around with trophozoites during the amoebic dysentery. So it's actually three. It is erythrocytes, mucosa, and submucosa of colon. Please go back and go through the life cycle of entamoeba. That is why I discussed entamoeba. See, now there's a question related to that, right? So the answer is C. The active form is called trophozoite. So if you knew the life cycle, you could have been able to answer this question, right? The trophozoites are excreted with blood and invade the mucosa and live in the submucosa of the large intestine. So they feed on these three things. All right, clear? Let's move on then. Question number 9. Infection of Ascaris usually occurs by, and this appeared in NEET 2013, A, CC fly, B, mosquito bite, C, drinking water containing eggs of Ascaris, and D, eating imperfectly cooked pork. So infection of Ascaris usually occurs by drinking water containing eggs of Ascaris. Also, I have again discussed the life cycle of Ascaris. Please go back and see that. Answer is C. Now, Ascaris is caused by consuming contaminated food, water, and raw vegetables. Now, there's one more disease. Yeah, by consuming undercooked pork can also result in infection. Now, there are two pathogens which reside in undercooked pork, raw pork, pig meat. That is when you consume that. Okay, the first one is trichinosis. Okay, the first diseases that occurs when you consume undercooked pork can result in trichinosis. Okay, it is caused by the worm Trichinella Spiralis. Okay, and the life cycle exists in one host, either it's man or pig. Okay, and man is like the end result. Okay, of that. The larvae remains in the striated skeletal muscles of the host, which provides it optimum conditions to grow and within 20 days undergoes encystment. Okay, the encysted larvae can remain for months to years and eventually calcifies and dies. The patients develop nausea, vomiting, diarrhea, and abdominal cramps. Okay. The treatment include administration of mebendazole. Okay, so if they ever ask you a question like which disease is caused due to consumption of undercooked or raw pork, you know it's trichinosis. There is also one more disease. We'll come to that. Okay, it is caused by the worm Trichinella spiralis. Now, consuming raw or undercooked pork can also result in infection from Taenia solium. Okay, so you have trichinosis, you also have Taenia solium. Okay, and the definitive host is man and the intermediate host is pig. Now, the encysted larval stage, Cysticircus cellulose. Okay, now cysti, they also in some textbook they write cysti. It's also called Cysticircus cellulose is what is consumed by humans. Okay, it causes the infection. So it's consumed by humans and which hatch out in the small intestine and mature in 2 to 3 months. Okay, the gravid segments pass out with feces in chains of 5 or 6. The segments are eaten by pig, and in the duodenum, the embryo 4 of the eggs rupture and liberates oncospheres and enters the circulation. They mature into bladder worm known as Cysticircus cellulose, which enters humans by consumption of raw or undercooked pork. Okay, so it remains in the pig, and if you eat the pig, if it is not cooked properly and it is raw, you can consume that into your body, okay, into your system. So it is very important that you cook the pork properly before consuming it. Okay, so basically Cysticircus cellulose, if they ever ask you the infective stage of Taenia solium, you know it is Cysticircus cellulose. Okay, now infection leads to abdominal discomfort, indigestion, diarrhea, and loss of appetite. Okay, So I have basically answered the question. This is just extra information to answer to elaborate on point D. But to answer the question, yeah, for question 9, it's C. Okay. All right. Let's next question. Okay, so question number 10. Identify the site where Wuchereria bancrofti is normally found in human body. It's appeared in Karnataka NEET 2013. Uh, we already discussed Wuchereria bancrofti in much detail. I even spoke about the life cycle. A, muscles of the legs, blood vessels of the thigh region, skin between the fingers. No, it's lymphatic vessels of the lower limbs. Okay, so the answer is D. Okay, so please go back and watch that part again on Wuchereria bancrofti. So it is found in the lymphatic system of the lower limbs. Okay, next question. Question number 11. Motile zygote of Plasmodium occurs in, this appeared in 2012. Motile zygote of Plasmodium. Again, we have already discussed the life cycle of Plasmodium in much detail. That is why I said it is very important to understand the life cycle so you can answer these kind of questions. Okay. So it's A, gut of female Anopheles, B, salivary glands of Anopheles, C, human RBCs, D, human liver. The motile zygote, okay, it's an ookinete, right? It is found in the gut of female Anopheles. So the answer is A. That is why it is important to understand the life cycle. So motile zygote is called ookinete and enters the epithelial lining of the stomach in the female Anopheles mosquito. Okay, clear? Simple. Like I said, if you knew the life cycle, you can answer questions like these, right? So that's why I discussed the life cycle of some of these pathogens. Let's move on. Question number 12. Vidal test is carried out to test. We'd already discussed this same question. This appeared in 2012. It's typhoid fever. Okay, so the answer is D. Vidal test is used to carry out tests for typhoid. We already discussed this similar question. Let's move on then. Question number 13. Common cold differs from pneumonia in that, and this appeared in 2012. A, pneumonia is a communicable disease, whereas the common cold is a nutritional deficiency disease. No, that's incorrect. B, pneumonia can be prevented by a live attenuated bacterial vaccine, whereas the common cold has no effective vaccine. No, that's incorrect. Pneumonia is caused by a virus, while the common cold is caused by the bacterium Haemophilus influenzae. No, that's incorrect. Pneumonia pathogen infects alveoli, whereas the common cold affects nose and respiratory passage, but not the lungs. Yes, that is correct. The answer is D. All right. So the pneumonia pathogen infect the alveoli and bronchioles of lungs, and hence there is no effective exchange of air. We already discussed pneumonia. Pneumonia vaccine is inactivated, killed bacterial vaccines. Okay, so they are inactivated vaccines, not live attenuated vaccines. And common cold, or also known as rhinitis, is caused by rhinoviruses. Okay, and infect the nose and the respiratory passages. Okay, so that's the correct answer for that. All right, so let's move on. Okay, so question number 14. Where will you look for the sporozoites of the malarial parasite? It's appeared in 2011. A, saliva of infected female Anopheles mosquito. B, RBCs of humans suffering from malaria. C, spleen of infected humans. D, salivary glands of freshly molted female Anopheles mosquito. Where you look for the sporozoites of the malarial parasite will be the saliva of the infected Anopheles, a female Anopheles mosquito. Okay, so sporozoites are the infective form of the malarial parasite, and they're present in the salivary glands, or they are ejected through the saliva. Okay, so if you check the saliva, you'll find sporozoites there. So the answer is A. Again, please go back and read the discussion on, listen to the discussion on life cycle of malaria, Plasmodium, that is, and you'll be able to understand this better. So sporozoites are the infective form of the parasite and are found in the salivary glands of the female Anopheles mosquito. So hence they are present in the saliva. Okay, next question. Question number 15. Which one of the following options gives the correct match of a disease with its causative organism and mode of infection? This appeared in Mains 2011. Disease, causative organism, and mode of infection. So you have A. Typhoid, Salmonella typhi with inspired air. No, that's not the mode of infection. Pneumonia, Streptococcus pneumoniae, droplet infection. Yes, that's possible. Elephantiasis, Bucharest bancrofti with infected water and food. No. D. Malaria, Plasmodium vivax, bite of male Anopheles mosquito. No, that's incorrect. So the answer is B. That's the appropriate answer. We already discussed pneumonia in detail. This caused by Streptococcus pneumoniae is transmitted by droplet infection. Also, Haemophilus influenzae also causes pneumonia. Right? So that's the correct answer. Let's move on then. Question number 16. Common cold is not cured by antibiotics because it is. This appeared in Mains 2011. We already discussed common cold. Common cold is caused by a virus. It's not an it's not bacterial. So you take in antibiotics for that. Okay, so clearly A is the right answer. Not much to discuss here. So common cold is caused by a virus, and hence antibiotics will not be effective in treating it. Clear? Let's go to the next question. Question number 17. Ringworm in humans is caused by, this appeared in 2010, it's caused by fungi. Right? We've already discussed ringworm in detail. Go back and listen to the discussion there. So the answer is B. Okay. Let's go to the discussion. Ringworm is caused by fungi. It's called Taenia. Okay. The genus is Taenia. So that causes ringworm disease. Okay, question number 18. Vidal test is used for the diagnosis of, again, the same question. We already discussed this. This appeared in 2010. It's A. Malaria, B. Pneumonia, C. Tuberculosis, and D. Typhoid. So the answer is typhoid. Vidal test is used for diagnosis of typhoid. Okay. All right. Let's move on to the next question. Question number 19. A person suffering from a disease caused by Plasmodium experiences recurring chill and fever at the time when, this appeared in Mains 2010. A. Sporozoites released from RBCs are being rapidly killed and broken down inside spleen. B. The trophozoites reach maximum growth and give out certain toxins. C. The parasite, after its rapid multiplication inside RBC, rupture them, releasing the stage to enter fresh RBCs. D. The microgametocytes and megagametocytes are being destroyed by the WBCs. So you already discussed this. The recurring chill and fever, that one that appears in the early afternoon and undergoes this, stage of cold stage, hot stage, and then sweating. Okay, that happens when the RBCs rupture and the Plasmodium pathogens, they infect new RBCs. Okay, so the answer is C. Okay, the answer is C here. All right, let's go to the discussion. So the recurring chill and fever due to merozoites which are released from the RBCs, the erythrocytes, and infect new RBCs. All right. Okay, let's move on then. Question number 20. Which of the following is a pair of viral diseases? This appeared in 2009. A. Common cold, AIDS. B. Dysentery, common cold. C. Typhoid, tuberculosis. D. Ringworm, AIDS. Okay, so which is a pair of viral diseases? It's clearly common cold and AIDS. Both are viral diseases. Okay, so the answer is A. Alright, so here I made a list. It's a table here. So AIDS is caused by HIV. Typhoid is Salmonella typhi. That's a bacteria. Dysentery is either amoebic or bacillary. Okay, so you have Entamoeba histolytica, and there are other bacteria also that cause dysentery. Tuberculosis, Mycobacterium tuberculosis, ringworm Taenia corporis, and common cold is a rhinovirus. Okay, so learn these causative agents. Like I already said, learn the disease, the causative agent, whether it's a bacteria, virus, protozoa, whatever the case, it's a ringworm, fungus, sorry. and it's a nematode, whatever the case is. All right. Okay, so question number 21. Match the disease in column 1 with the appropriate items, its pathogen or prevention or treatment in column 2. So, Amibiasis, Treponema pallidum, use only sterilized food and water. DPT vaccine, use oral rehydration therapy. So, Amibiasis, use only sterilized food and water. Diphtheria is DPT vaccine. Cholera is use oral rehydration therapy. Syphilis is Treponema pallidum. So you have A2, you have B3, you have A2, B3, C4, yes, syphilis D1, yeah. So the answer is B. Okay, this appeared in the year 2008. Okay, so amoebiasis can be prevented by using sterilized food and water. DPT vaccines used to prevent diphtheria. It's also used to prevent pertussis, that's whooping cough, and tetanus, so that's what's DPT. Cholera is treated using oral rehydration therapy, and pathogen for syphilis is Treponema pallidum. Alright, okay, so we've answered the question here, pretty easy. Then let's move on. Okay, so we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture, and I hope you've learned something new today. And please don't forget to like, share, and subscribe to MG NEET Biology. And I'm available on Facebook, Twitter, and Instagram, so you can follow me there for more updates. And I'll see you in the next video lecture, right? So bye.